Talk a little bit more about this whole electron transport thing. These electrons have a lot of energy, and in moving them from one place, one location to another, a little bit of energy gets liberated every time that happens. And we could just take our molecule. Remember, we're using glucose. We could use a protein. We could use a fat, but we're going to just choose to use this monosaccharide for a simple example, knowing that there are a lot of other foods and macromolecules and monomers and such that get digested. We're just going to focus on this one monosaccharide. And we could release it all at once, but if we release it all at once, we get explosive release of energy that's not helpful because we're not going to be able to harness that. So instead, what we're going to do is we're going to release a little bit of energy in a series of steps. And so every step along the way, as an electron gets passed from one carrier to another, it will be losing a little bit of energy along the way. And so cell respiration, the process of taking your food and turning it into an energy source that your body can actually use, is not a one-step process. There's lots and lots of little steps to that. And so today we're going to look at a couple of those steps, starting with this thing called glycolysis. And when you look at these steps, they are color-coded. They are color-coded in your book by the stage name. So glycolysis has got this blue color to it. Red is going to be for pyruvate oxidation and the citric acid cycle, and oxidative phosphorylation, which we will not get to today, will be color-coded as purple. When you look at a cell and you look at where this process is taking place, or all these different processes, I should say, because there's lots. It's like this entire thing here is the cell. And then we've got a really, really blown up, what is this guy here? Mitochondria, yeah. So I've got a really blown up mitochondria. Obviously, mitochondria aren't, half of the cell size. We're just blowing it up for emphasis here. And then what we're going to focus on today is we're going to be looking at this glycolysis, which is not taking place in the not taking place in the mitochondria, but instead it's taking place in your cytoplasm, your cytosol. And then depending on how much time we have, we may go into the second stage, which is this little guy here. We'll just play that by ear and see how we do. But there are these steps. We've got glycolysis. We've also got our red steps here. And we've got our purple steps. But today we're just going to focus on our blue glycolysis. On this slide, we can also see that there is this reference to something called substrate level. Actually, they just, wrong way. I'm going to have to get rid of some of my circles, I think. Don't look right anymore. Uh-oh. Let me clear the screen. Yeah. I've got an ATP, and then underneath it, I have substrate level for those first two stages. That is referring to how the ATP gets made. And there are two ways that ATP can get made. We're starting with a DP. And that ADP is going to be converted back into a molecule of ATP. So it's going to have a what added onto it? Phosphate group. Yeah, we're going to add a phosphate group onto it. There's two ways to add on that phosphate group. One is through what they call substrate level phosphorylation. And that word is here, in case you were, oh my God, I don't know how to spell that. There it is. And that's the second bullet point there. Substrate level phosphorylation is where, as soon as a phosphate gets released from a molecule, it gets transferred back on to the ADP to become ATP. It happens in glycolysis, as well as that second stage that we'll talk about, citric acid cycle. There's very little ATP that's formed that way. The bulk of the ATP is not formed by substrate level, but by something called oxidative phosphorylation. And I'm not going to explain oxidative phosphorylation right now because it's going to take an entire period. It's complicated, and it is the last step in cell respiration. Okay, so we're going to start off gradual here, and we're going to do glycolysis first, and then eventually we'll get to that last stage, which is the oxidative part, where you can make almost all of your ATP is made in that step, but that's the very last step. This image is showing that in substrate level phosphorylation, there is an enzyme that is catalyzing the conversion of that phosphate back onto the ADP to become ATP. And it's a one-stop shop. There's one step, phosphate gets added on, and you're done. That's nice and simple, but it's also the least efficient, and you don't get much energy that way. So although we do it, it's not as important as the other ways that we generate ATP, which is the oxidative one we'll talk about later. In total, when we take one molecule of glucose and we take that and we completely oxidize it. What do I mean when I say I completely oxidize a molecule of glucose? What am I getting rid of, or what am I removing? Electrons. I'm removing the electrons. Okay. So when I say oxidize, remember oxidize means to take away the electrons, and something else is going to become reduced. Okay. So we're going to reduce something else is going to take those electrons away. Something else is going to pick up those electrons. It's not a lot of ATP. And we'll talk more about that later on, but the basic take-home message from this is that much of the molecule of glucose gets wasted, quote, and put that in quotes because it's not really wasted, as heat energy. It's still energy. It's just not a form our body can really use. I mean, but it sort of does. I always kind of get never quite sure to say if it's really wasted or not, because I mean, obviously, if we didn't have that heat, that wouldn't make us warm, and that would threaten our survival, right? If we weren't warm enough, then we wouldn't be able to perform all those chemical reactions that require a certain temperature for those enzymes to work. Remember, those enzymes are working at a particular temperature. So if we didn't release that heat, then we wouldn't have that ability for the enzyme to work. So even though it goes away and we have to keep generating more heat, it is important that we generate that heat. All right. So let's talk about glycolysis. Actually start with 9.2. Glycolysis is the process of harvesting energy by oxidizing glucose. So again, we're liberating some of those electrons. We're taking those away. We're stripping the glucose of it. And in doing so, we're going to come up with a different molecule that's called pyruvate. And that it will be the endpoint of glycolysis of this series of reactions will be the formation of pyruvate. Glycolysis refers to the word, or means, glyco means sugar, and lysis means to split. So what we're doing is we're splitting glucose. How many molecules? What's the formula for glucose? You guys sound like you're in church. And so we're trying to think of a phrase. Phrase that we'd say, amen. C6H12. That's glucose. Okay. And what we're doing is we're going to break that down into two smaller molecules. I don't know the formula for pyruvate. But basically, we're making our glucose into 3 carbon molecules. So we take it and split it down the middle, and now we've got 3 carbons rather than having our 6 carbons. But we have 2 of them. We have 2 molecules of pyruvate. Glycolysis has 2 parts to it. First of all, it occurs in the cytoplasm. Remember that, because that's going to be important for various reasons. And I like to use an analogy for this one. There's an energy investment stage and an energy payoff stage. And the idea here is you have to put something in, you have to put a little bit of energy in to get it going. And if you don't put that energy in, then you won't get more energy out. Okay. Anybody do fundraisers? Do fundraisers? You buy something cheap, right? And then you sell it for a higher price, right? And you make a profit, and that goes for your fundraising. Okay. Same thing with glycolysis. What you're doing is you're putting a little bit of energy in to jumpstart that, and then you'll get more energy back out as a result. Okay. All of this happens regardless of whether or not oxygen is present. And that is also very important. Happens whether or not oxygen is present. So this is the only stage of producing energy that our body does that does not require oxygen. And it's also the least efficient. So we can produce very little energy with glycolysis. And that will also become apparent and important down the road. So here we are, we're out in the cytoplasm. Our glycolysis is converting some of our glucose into ATP. By what method? It's phosphorylation. Which kind?
Oxidative.
Substrate.
I just looked at the oxidative.
Yeah, that's later on. So we're going to do it by substrate level phosphorylation. Okay. We're not going to produce much of it. I think, if I remember correctly, come on, switch. There we go. There's at least 12 reactions that occur in glycolysis. You do not need to know those 12 reactions. This is kind of a breakdown of what's going on. And really a summary. I know it seems complicated. It's not too bad when you break it down a little bit. Let's just start at the top. We have an energy investment phase. So we're taking glucose and, okay, we have to invest something in it. So the first thing we do is we jumpstart it and we use a couple of ATPs. Okay. Now, this is the investment, but this is our outcome. Our outcome after all of those reactions is that we create 4 ATPs. Woohoo! 4 ATPs. How many did we use? 2. So we actually only made 2 ATPs. Wah, wah, wah. Yeah, that's the whole lot of nothing. There are some organisms that all they do is glycolysis. So this is it. That's all I got. But here's kind of an important bit here. We also make, what were those NADHs that we talked about? Remember those? What were they carrying? Yeah, I heard it. Electrons. They were carrying electrons. There are taxis, or shuttles. These NADHs are kind of gold. I just want you to be aware of those NADHs and that they were made, as well as these hydrogen ions were also produced. These little guys here. Because both of those things are going to become important when we get to the last part of cell respiration, which we won't talk about today. Okay, so these guys are going to taxi electrons. They've captured the electrons. They have now become what? What's the term called? reduced. They've become reduced. They started off as being oxidized. They were NADs, they were NADs, and now they are reduced to form NADHs. And they're going to carry those electrons to another part of the cycle of cell respiration. The other thing that we make along the way is our molecules of ATP. Sorry, not ATP, pyruvate. Okay, so I've taken that sugar, I've taken my glucose, and I've split it in half. And I made it into 2 3-carbon molecules rather than 1 6-carbon molecule. Don't worry so much about numbers. College Board, as well as me in particular, are not going to be worried about how many molecules of ATP are made during glycolysis, how many are used during to jumpstart it. It's more a matter of understanding that there's not very much. The thing is, depending on the conditions, the environmental conditions, the organism, different species, you get slightly different values in terms of how much ATP is produced and how much is used. So don't focus on that. Focus on the big picture, which is in glycolysis, we're producing some ATP. By what process? What kind of phosphorylation? Substrate level phosphorylation, but not many. We're only producing, in the end, our net here, we're only producing 2 ATPs. So it is not very efficient at generating energy from this process, which is okay because we'll make it up along the way. But it's not important that you know the values. It's important that you know what came in, which was glucose, what comes out, which is pyruvate, where it took place, cytoplasm, and what you made. Again, not focusing on the numbers. You made ATP, you made NADH, and you made pyruvate. Questions on that? All right, so pyruvate, now we have 2 molecules of pyruvate. And those molecules still have a lot of energy in them. But in order to do anything with that energy, we have to do kind of a intermediate step. And that's only going to happen. It's only going to proceed to the next step if there is oxygen. So we have to have oxygen present. If we do not have oxygen present, we can't finish this whole cycle with the pyruvate. And a lot of that pyruvate is going to be lost in terms of its energy value. It will be lost. But if we assume we have oxygen, or I shouldn't say if we assume, if we have oxygen, we can proceed and take that pyruvate. And again, we're going to oxidize it. What did I say that meant?
Electron. Take away electrons.
Yeah, goodbye, electrons. You're gone. Electrons are gone. They're going to go away. So before the next step in the process can happen, there's like a little mini step that has to happen. And that's called oxidation of pyruvate. The next step is going to be called the citric acid cycle. But we can't do that until we get a different molecule, because that pyruvate molecule won't work with citric acid. Instead, it has to be something called acetyl-CoA, this guy here. And to get to this acetyl-CoA, we have to get rid of those electrons, some of the electrons from pyruvate. And in doing so, I said that pyruvate was 3 carbons long, but we're going to split it again, and we're going to release carbon dioxide into the air. So this is one of the molecules of carbon dioxide that we breathe out when we're breathing. It's coming from the splitting of the pyruvate into this acetyl-CoA thing. We oxidize the pyruvate, so we're giving up electrons, and the electrons are going to go to what molecule? NADH. NAD is going to get reduced again, different molecule, another taxi. It'll pick up another couple of electrons. And in the end, bada boom, bada bing, we make acetyl-CoA. This has to happen in order for everything else to happen after this. And it will only happen if there's oxygen. No oxygen, and everything shuts down. And you've all experienced this. I experienced this yesterday probably 100 times for 45 minutes. Anybody heard of cyclocross? No? Yes. So cyclocross is this crazy bicycle sport where you are in a park and you're riding your bike over grass and mud and gravel. And occasionally you have to pick your bike up and actually jump over barriers. And your goal is to beat everybody to the finish line. It's a lap and you do it repeatedly. And your goal is to try and get to the finish line before anyone else does. So people will go out too hard and then they'll start to fade and then you have to catch them. Anybody do like cross country? Anybody have that happen in cross country where people go out like really fast and then you catch them? No. Anyway, so I was doing this and I went out too fast. Sad face. And when you go too fast, what happens? You get tired because you don't have any energy and you don't have any oxygen. You can't breathe, right? You can't breathe. You can't breathe in enough oxygen and get enough energy from that oxygen. Because what have I done when I'm not getting enough oxygen in my lungs? What is the only stage of cell respiration I'm going to be able to do? Glycolysis. And how much energy am I producing? Not much. Okay, so you have to slow down, right? So my whole race was slowing down, getting more oxygen into my lungs.
Did you win?
Speeding up. I'm a winner in my own mind. How's that? I did better than I thought I would, so I'm excited. I hadn't raced in 4 years, so I was excited. But this is what happens if you don't have that oxygen. This oxidation of pyruvate cannot occur. Okay, and we've all had that circumstance where we start to get short of breath and we have to slow down from whatever we're doing. How about running the mile? You run the mile? In gym, you have to run the mile? No, you don't. They don't make you do that anymore. Pacer test? You ever go out too fast and you're like, oh, I gotta slow down. I'm never gonna make it. Oh, okay, speed wagon. Okay, that didn't work. Okay, that's our next step. Is that oxidation of pyruvate? 2 more slides and then we'll stop. So now look where we are. We're in that red spot. Where are we in the cell? We're now in the mitochondria. Oh, so we have gone from cytoplasm and now we're in the mitochondria and we're going to stay in the mitochondria. Here's my pyruvate. My pyruvate is going to get transported into the mitochondria. I'm going to lose a carbon dioxide. I'm going to reduce my NAD to become NADH. I'm also going to pick up a proton, and H plus is a proton. And I'm going to make this molecule called acetyl-CoA. Those are my 3 steps in oxidation of pyruvate. That has to happen because this molecule is needed to go to the next step. If I don't have that, all this shuts down. And none of this is going to happen if I don't have oxygen. All right. Questions?